Take it
Great.
away.
Well, thank you so much to the organizers for inviting me to present today. So I'll be speaking about human-plant interactions and the ecology and evolution of infection. So to start off with, these are just a few examples of plants that you may be familiar with. These are not only beautiful in nature, but they're also sources of some of our really important medicines that have transformed the way that we manage pain and treat cancer and deal with heart disease today. In the world, we have around 374,000 plants on Earth, and out of those, roughly 28,000 have been used by humans in different forms of traditional medicine. And yet, when you look at that great biodiversity, we really have only barely scratched the surface. Of the ones that we have looked at, many of these have become key components of our essential systems of medicine. These are some examples from the World Health Organization's list of essential medicines of therapeutics that every hospital should have. And as I mentioned before, these are important in the treatment of cancer, infection, which is where my interests really lie, heart disease, pain, and so on. Now, medicinal plants are important not only to the Western consumer in the pharmaceutical industry, but also to the majority of people that live on this earth. In fact, they compose the primary pharmacopoeia, or the primary source of medicine, for any between 70 to 95% of people that live in developing countries. And so people are actually going out there wild harvesting or cultivating plants as their main source of medicine. And so developing a better understanding and appreciation of their efficacy and their safety is really important, not only for future medical innovations, but also in delivering better care to these large populations. So as an ethnobotanist, I have the great joy in a normal year of going out and traveling to biodiversity hotspots in the world where we look for plants that have been used in these different systems of medicine. In yellow, you can see some of the different spots where we have collection sites. And this is through a vast network of collaborations that we've established in different countries with professors across the globe. Now, you may be wondering, I haven't identified what ethnobotany actually is yet. Let me just define it for you. Ethnobotany is the study of how people engage with plants. So this is not restricted only to medicine. It also includes the use of plants for everything from food to building materials and musical instruments, art, and so on. And ethnobotany has also been defined as a science of survival. So this is really the field where I bring together not only an understanding of these human nature interface interactions, but also how that relates to studies we do that. To go through this kind of work requires a lot of missions, a lot of ethical guidelines that we follow. So we have prior informed consent when we collect data on the uses of plants. We follow international codes of ethics. We follow conventions like the Convention on Biological Diversity and the Nagoya Protocol regarding access and benefit sharing of ingredients that are studied. And then we also have extensive permits and international collaborative agreements. My lab is also a USDA inspected facility, so we're able to receive plants and fungi from anywhere in the globe to study in the group. Our general approach is to basically reverse engineer traditional medicine to not only understand if and how these products work, but also to yield chemical blueprints, again, for innovations in the antibiotic space. So we start with ethnobotanical information through interviews or through analysis of historic records. We identify those plants, we rip out the chemicals, and again, each tissue of a plant may have hundreds, if not thousands, of unique molecular entities. We then subject them to biological assays, followed by iterative steps in bioassay guided fractionation, and then go through these steps of analytical chemistry to identify the various compounds that are responsible for their activity. Our outputs, or our goals, or areas of focus are really around the discovery of antibiotic agents in the classic sense. I'm also interested in antibiotic resensitizing agents, anti-virulence therapeutics, including those that target both biofilms and toxins. In total, this resource is now composed of over 2,000 extracts derived from 600 species, and it's unique in the world for its focus on plants and fungi used by local people for the treatment of infectious disease. And again, this is because it's strictly inspired by traditional medicine, but driven by bioactivity. So there are kind of four elements of our research approach. We have the field research aspect. This is a photo of me from a year or two ago in Albania. We have the herbarium, which is a kind of like a dead plant museum where we collect a biological repository of all the specimens that we work with. The phytochemistry lab, where we look at the chemistry of these extracts, and then the microbiology lab, where we test them for activity in a number of different pathogens. We're focused on the main AMR threats. So this currently includes crab or carbapenem-resistant Acinetobacter baumannii. We also work on pathogenic fungi, including Candida auris, and a number of other bacteria as well. We've begun work on pseudotype models for COVID as well, and are starting our initiative to screen this unique collection for potential inhibitors of viral entry. So what do we know, though, about antibacterials or strict antibiotics from plants? Well, the truth is we don't really know that much. No one's ever conducted a systematic review of the literature when it comes to this question. So we took this on during our kind of pandemic review writing period. And we looked at all of the papers published since the 1940s until recently, and we're able to identify 6,000 articles on this topic. Though with further analysis of the top papers, we found that, and just looking at the top 66% of those, we found that only 16% of these publications actually meet our criteria for looking at antibacterial activity of plants in a really rigorous way, following established protocols for the detection of an MIC value. In total, though, we found 958 plant species and 468 compounds that have been assessed. So what are we doing in my lab on the research side? We're looking at different examples from traditional medicine to identify or validate whether or not different historic remedies could work in the management or treatment of infection. We have looked at, for example, at plants that were used during the period of the American Civil War for wound therapies, and found some efficacy in a few of those species in terms of growth inhibition and biofilm inhibition. We've also taken a look at much older documents, including the Ben Cao Gang Mu, which is a traditional Chinese medical text that goes back to the 1600s. And we found some interesting uses, historic uses of ginkgo biloba that actually panned out as having some antibiofilm efficacy. We've also looked at some of our work, our current work in the Balkans, where they use plants like St. John's wort as a skin ointment or liniment for treating skin disease, and here also found some interesting activities in terms of both growth inhibition and biofilm inhibition, depending on the ways in which the St. John's wort was formulated. As a point of information, this plant actually contains a number of phototoxic compounds that can lead to severe skin reactions following exposure to the sun. But interestingly, this traditional preparation, which involves steeping the flowering parts of the plant in an oil base in the sun for a number of days, actually removes those phototoxic compounds. So some of our limitations in this kind of work, of course, is the ability to isolate the active compounds. So we recently published just this year the design for an in-house built robotic platform for the isolation of compounds from plants. This is literally made with Lego pieces from my kids' home set and some hardware from just a regular hardware store. And so we were able to produce this for under $500. A large open bed fraction collector of this type can cost up to $20,000. And so we felt this was really valuable, not only for our own work, but also for others in this field that need cheaper options for isolation of compounds. What this enabled us to do is it enabled us to actually isolate larger amounts of pure compounds from the plants. And again, some of the active constituents may be present at only levels of a few percent or less than 1% in some cases. But we were able to isolate enough of these compounds to actually achieve crystalline structures, and we're able to determine full conformation with X-ray crystallographic experiments, in addition to our mass spectrometry experiments and NMR analyses. Some of these have resulted in a number of different compounds that we've identified to have some interesting antibacterial activity. I'll just give you some examples. This one is pentagalloyl glucose. It is a compound found in the leaves of a number of plants found in the Anacardiaceae family, or the poison ivy family. In this case, we isolate it from a plant known as the Brazilian pepper tree. What caught our attention is that this compound actually has some pretty good activity when it comes to preventing the kind of resistance creep that you typically see when you expose Abomani, or really any other microbe, to chronic low doses of an antibiotic. Here's an example of the creep, or the MIC creep, that you see with tetracycline, and this is in the presence of our compound that we isolated, pentagalloyl glucose. It's also not very toxic to human cells. There are some limitations with this compound because it does have poor bioavailability, but there are some potential avenues that we're exploring with medicinal chemists to see if this could also be optimized or perhaps leveraged as a topical therapy for wounds that are infected with Abomani. Another plant that we've looked at is that of Calocarpa americana, or the American beautyberry. In this species, we identified this claridine diterpene, and this was part of our campaign to identify antibiotic resensitizing agents, or compounds that can restore the activity of otherwise resistant microbes. In this case, we use MRSA, or methicillin-resistant Staph aureus, and through our checkerboard analyses, we were able to identify this compound out of a large mix of hundreds of molecules, and determine that this works. It works quite well with oxacillin, but also other beta-lactams, including meropenem. You do not see the synergistic effect, though, with vancomycin. So this is a paper that was just recently published as a note in ACS Infectious Diseases that you can check out, and we're currently working with a team of medicinal chemists at Notre Dame to generate synthetic analogs of this, to further use this as a tool compound to understand better what the potential mechanism of action is. Other targets that we've looked at include those for virulence and biofilm formation. I've been drawn to these particular targets because of the things that we've found through analyzing plants used in traditional medicine. There have been a number of examples where I've seen the consistent use of a plant for traditional treatment of a skin infection, and yet that plant has no classic antibiotic activity. What we've found instead is that these plants turn out to be quite good sources of anti-virulence compounds. Here's an example from the European chestnut. We've identified some novel hydroperoxycycloargines in the chestnut leaves that block a quorum sensing system in staph known as the accessory gene regulatory system. And this, in the end, results in attenuation of dermonecrosis and production of a large suite of toxins. Likewise, we just published this year the discovery of some triterpenoid acids, again from pepper tree, and these also block virulence. You can see that while in vitro they all performed quite similarly, there was one in particular, compound number 2, that was able to show some really nice prophylactic activity in blocking dermal necrosis in an intradermal MRSA injection model in mice. In addition to virulence toxin pathways, we've also looked at the ability of certain plant compounds to block biofilm formation and also biofilm maintenance. Here's one example we've done with the roots of the elm leaf blackberry, where we found there was actually a mixture of ellagic acid glycosides that showed some really nice activity in blocking the adhesion steps and accumulation of biofilm. We've worked with chemists to generate individual ellagic acid glycosides for study, and found that while one particular sugar derivative was most effective, none were as effective as the original plant extract, which is made up of this mixture of these compounds. So in conclusion, and as eloquently put in the previous talk, antimicrobial resistance is going to continue to be a threat, even during the time of COVID, while we're facing large numbers of morbidity and mortality. We still have over 700,000 people each year dying from otherwise drug-resistant infections. We need new approaches to therapy, and this is where I think that plants can really play a great role, both in looking for novel antibiotic scaffolds, but also new ways of developing these adjuvant approaches through the use of biofilm inhibitors or virulence inhibitors and so on. And again, if you're a student looking for an area to research, I mean, there are literally 28,000 plants that still need to be studied. So this is an area where it's a bit difficult to become scooped. There's a lot of biodiversity, a lot of chemical diversity out there to explore. And in many cases, these plants are actually under threat due to factors such as globalization, expansion of human settlements, and also climate change. And so there is a sense of urgency also to evaluate these resources before we lose them. So lastly, I want to thank our amazing research team. I have a great group of collaborators in the US and beyond. I want to recognize our funding agencies as well. And I'm going to put out a little pitch. I have a podcast. It's now in its second season. If you're interested in learning more about the ways that plants play a role in health through the food medicine continuum, you can check that out. It's available on all the major podcasting services. And that's it.